Hello and welcome. Today we are going to discuss the immune system, and we're particularly going to concentrate on the innate immune response. So we're going to look at the immune system as a whole. There are two types of immune defenses. When we speak about the innate or the non-specific, it's going to have very important different functions than the adaptive or the specific. The one or two big differences are in the innate or the non-specific immunity. The immune cells are unable to recognize the specific invaders. They don't have anything to help them know this is this virus or this is this bacteria. They cannot recognize specific invaders, and they cannot keep memory of this invader. They can't react quicker on a second launch because they have no memory of the invader. The adaptive or the immune has both of these. Those cells that are activated can recognize specific antigens, and they will keep those antigens in memory for a second quicker and more effective response. So let's look at those two arms of the immune system. The innate response is what we are born with. So we're going to see the first and the second line in a minute. And when we are born with those types of immunity, the response is going to be very fast, almost immediate. In the adaptive immune system, we're not born with those defenses. As we grow and as we get exposed to different antigens and bacteria, we will acquire this immunity. So understandably, it's going to take a little longer to develop. When we talk about the innate immune response, we have a first line of defense, and it consists of the layers of skin. It consists of the membranes on our body that make the barriers and everything that is secreted by those membranes. We call this the first line of defense. So it keeps the invaders from coming in. If this barrier is broken and the invaders come in, we have the second line of defense here, and it's still non-specific. So we're going to be talking about certain cells, the macrophages, the NK cells, certain chemicals, certain responses in the body. So we call this the first and second line of non-specific immune response. The third line will belong to the specific. We're going to talk about this in a different lecture. So let's concentrate on the first and the second line of defense. This is the first line of defense. So we are looking at a human being here, this little baby. We're going to look at the membrane. So when we look at the intact skin, it is a physical barrier against entry of microorganisms. Many other things in the skin, the secretions formed by the skin, the sweat, the sebaceous secretions. When we look at the different glands that are in the body, the tears, the saliva, the different secretions, they contain certain antimicrobial agents. The lining of the membrane, so the mucous membrane that is lining the stomach, the digestive tract, the mucous membrane that is lining the respiratory tract, again, they are a physical barrier to prevent microorganisms from penetrating, but they also produce secretions. So as an example here, the hydrochloric acid in the stomach, highly acidic, destroys almost all organisms. The urine in the urinary bladder is highly acidic, and so forth. Not to forget the respiratory tract. In the upper respiratory tract, we have the mucociliary escalator, which is in the lining epithelium of the respiratory membrane. We have the pseudostratified columnar ciliated epithelium, which has the cilia. we have the goblet cells that secrete mucus, and any inhaled toxins or antigens are going to stick to this membrane, and the cilia can beat and bring any inhaled particles up to the upper airways, where we can cuff them and get rid of them. The second line of defense, we're going to talk about several different entities, either cells or group of cells that do certain functions. So we're starting with the phagocytic cells. What are phagocytic cells? A group of cells that do the function of phagocytosis, and these are some examples. The neutrophils of the white blood cells, the macrophages, which are activated monocytes that go to the tissues, dendritic cells in the skin and in other areas. All of these are phagocytic cells. What do they do? Phage comes from eating mouth, so they actually take in the microbes and they eat them and they process them, and this is the process of phagocytosis. So if this is a microbe and this is a phagocytic cell, it's going to be attracted towards the microbe, make processes that will extend around the microbe, pull it in, ingest it. It's going to attack it by means of a lysosome. The lysosome has digestive enzymes. It will digest the microbe. It's going to break it down, kill it, and any residue is going to be excreted outside the cell. So the process of phagocytosis actually kills and eliminates invaders. Eosinophils, although they are not phagocytic cells, but we had to put them with the group of cells, they are very important white blood cells. They secrete chemicals that particularly destroy parasitic worms. Another type of cell in the second line of defense is the natural killer cell, or NK cell. What is the nature of the NK cell? It is actually a lymphocyte. It's an unusual lymphocyte. It's a very big lymphocyte. It has granules, and when we look at it, what does it do? It has the ability of destroying cells. So remember, killer cell because it kills cells. What does it do? It's going to come very close to an abnormal cell or an enemy cell. It's going to start producing perforins. Perforins will poke holes into the wall of the cell, and then they will secrete digestive enzymes called granzymes. Those digestive enzymes will cause death and apoptosis of the cell, and the macrophages can come around and do the final cleanup. So remember, natural killer cells kill abnormal cells. Another group of proteins that help in the second line of defense is a group of proteins called complement proteins. I just wanted to give you a visual, a 3D of those proteins, so you remember proteins take a three-dimensional shape, and these are the shapes of some of those complement proteins. So what are they? They are circulating proteins in the plasma that circulate in an inactive form if there is no invasion in the body. But they will become activated if they will sense the need to do that. So how is complement activated? Complement proteins are activated through several pathways. We're going to look at specifically two. The first one is what we call the classical pathway. In the classical pathway, complement will see what we call an antigen-antibody complex. They will see an antigen and two antibodies attached to its surface. When they see this, they will stop. They will be activated to launch their immune defense. In the alternate pathway, Complement will see only an abnormal cell. They will recognize that cell wall does not look like the human cell wall. They're going to recognize it either belongs to a bacteria, a fungus. The cell wall is abnormal, and they will stop there and they will be activated. So this is the classical pathway. So complement, we give the complements the letter C. There are many of them. We're going to look at the classical pathway. So this is the antigen antibody complex. When there is an antigen and two antibodies attached to it, complement will recognize this is where I need to stop, be activated, and help destroy this antigen. What is complement going to do? Several of them are going to be activated in what we call a cascade. Cascade means a very specific order. So one will activate a couple more, and these will activate some more. We don't need to know the specifics right now of the different activation in process, but eventually at the end of this cascade, at the end of this activation of complement, complement will be able to perform three functions. Number one, opsonization. Number two, inflammation. Number three, cytolysis. We're going to look at these in a minute. So in the classical pathway, complement has to see an antigen antibody complex. In the alternate pathway, all it sees is an abnormal cell wall that does not belong to the human cell. Complement is going to stop and be activated. It recognizes this is abnormal. Again, there's a sequence of activation. Different members of complement will be activated, but the final outcome is going to be again opsonization, inflammation, or cytolysis. Let's see what these three mean. So this is opsonization. If complement is activated, opsonization means coating on the surface. So this is complement. It will coat the surface of the microbe, make it very evident, and now this will enhance phagocytosis. Phagocytic cells are going to recognize this is a microbe. They're going to come and they are going to destroy it. The second thing is complement will take part in activating the process of inflammation. How do they do that? They activate the cells that produce histamine, mast cells, for example, and histamine is going to produce, and inflammation is going to occur. And as we will know in a minute, inflammation is a first step towards healing. So inflammation will lead to healing. The third thing that complement can do is several of those members will adhere together and make one big complex. So these are several complements attached to each other, and they make a big complex. We call this membrane attack complex, MAC for short. And what is this complex going to do? It's going to attach to the surface of this abnormal microbial cell, and they will perforate or make holes in the wall of this cell, and eventually this cell will die and cytolysis will occur. So remember the three functions of complement. Opsonization, this will help phagocytosis. They will help in the process of inflammation, which will help healing. They will lead to cytolysis of an abnormal cell. Another member of the second line of immune defense is the interferon. If I were to ask you what is the function of interferon, The function of interferon is to prevent spread of viral infection. Let's see what is interferon and how does it do that. So to understand this, we need to talk a little bit about a virus. What's the virus? It is an invading organism which has to penetrate into a body cell, our body cells, and when it does that, when it comes in and penetrates, it's going to take over this cell and replicate itself, make many thousands and millions of copies of itself, and then it's going to destroy this cell, come out and invade the next cell, and so forth. So virus particles are invading particles. They enter a cell, they take it over, they burst out, destroy the cell, and they go to the next cell. The function of interferon is to prevent this from occurring. How does it do that? So if this is the first cell that is going to be invaded, this is my host cell. Once the virus enters in, this cell has the ability of making a protein called interferon. Interferon is going to leave the infected cell and go to the neighboring healthy cell. And it's going to stimulate this healthy cell to produce a protein. What's the name of this protein? Antiviral protein. What's the antiviral protein going to do? Give immunity to this cell, so that if the virus comes in and penetrates, the antiviral protein is going to completely block it. It can't do anything, can't take over the cell, cannot replicate. It is going to be completely paralyzed, and this cell is going to be spared. So this cell is protected, but who gives it the signal? It is the interferon. Interferon cannot help this cell. This cell will eventually die, but it will give immunity to all of the neighboring healthy cells, and accordingly interferon will protect the cells from the viral replication. So remember, who produces interferon? The infected cell. Who produces the antiviral protein? The healthy cell. The process of inflammation, as we were talking, the process of inflammation helps the healing of our body. So this is the story of inflammation. If there is an injury to the body, and over here this looks like Billy was skating and he scraped his knee, he has a big gash, there's bleeding, and there are microbes that have already entered through the skin. So the process of inflammation is going to start in this localized area of injury. What will happen? Immediately some chemicals are going to be produced in this area. We call these the inflammatory chemicals. The most important is histamine. What is histamine going to do? It's going to cause vasodilation of the blood vessels in this area of injury. Vasodilation means the diameter of the blood vessel is going to increase. If I open up the blood vessel, there's more blood flow into this area, which we need. So there's more blood flow coming to this area. Increased blood flow is called hyperemia. What does the increased blood flow do? It's going to bring more blood to the area, and it's going to, because of the vasodilation, there's going to be increased permeability from the blood vessels. So I am allowing things to permeate out of the blood vessel into the injured area. What specifically is helpful here? Phagocytic cells. So here we are. White blood cells are going to come and eat up and destroy all of those microbes. What else can permeate? Fluid will come up. So fluid will come up and bathe and clean up and dilute the toxins. So through this process, we are removing toxins, we are removing bacteria, and the process of healing is going to start. So when we look at an area of inflammation, so if we look at this area in a few hours, how is it going to look like? And this explains the cardinal signs of inflammation. Number one, the area is going to be red. Where does the redness come from? The increased blood flow to the area. It's going to be hot because, yes, the temperature of blood is a little warmer than the skin. It's going to be swollen. Where does the fluid come from? Increase in the permeability of the capillaries. Fluid will ooze out, and this area is going to be swollen. And pain comes from compressing on the nerve endings that are in all our body, and particularly the nerve endings of the skin. So these are the cardinal signs of inflammation: red, hot, swollen, and painful. Fever. Fever is an immune response to the body. How does fever occur and what causes fever? Fever is caused by a substance called pyrogen. Where does pyrogen come from? Pyrogen comes either from the white blood cells, the leukocytes, while they are launching an attack, they produce pyrogen, and sometimes they can be external, some toxins from some bacteria. So when pyrogens are in our system, they will go to the hypothalamus, and the hypothalamus is the thermostat of the body. This is where our set temperature is, and they will increase the set temperature of the body to a higher temperature as long as we are having a reaction or an infection or so forth. Once the infection goes down, the set body temperature goes back to the normal. Now, why do we say that this is an immune response? How does it help? At higher temperature, not too high, very high temperature can cause damage to cells, but a little rise in temperature will increase our basal metabolic rate. It will help the cells to work faster, and with this higher metabolic rate, there is more healing towards, there's more healing and overcoming the infection by our normal body cells. And this concludes the innate immune response of the body, and thank you.